Hello, welcome to our lecture on the scientific process. During this lecture, I'm going to give you an overview about how science in general works, and specifically how we use the scientific process to answer specific questions. We'll start with an overview of essentially how science works in a broad perspective, and then focus in specifically on how we use the process of science to collect data and to answer questions. The scientific process can be broken down in a number of different ways, but this is really the big picture about how science fits into the context of society. We generally think of science as being a process of testing ideas and then drawing conclusions from the results of those tests. But really, there's a lot of interaction between that core practice of science and a number of different facets of society. So one of the things that feeds these ideas that we want to test is the interplay between the information we've already determined from testing previous ideas and the new questions that existing information sparks. Frequently, existing data motivates us to go out and find more data on the same subject. We start asking more questions, better questions, and this feeds the process of exploration and discovery. Sometimes we'll discover brand new ideas, brand new questions to ask, which then feeds us more ideas that we can test. And so there's this constant feedback loop between discovering new information, testing ideas about it, which then raises more questions and leads to more exploration and more discovery. Now, once a set of ideas have been tested, they don't just automatically enter into the scientific knowledge base. Instead, those ideas are sent out for analysis by community members who have expertise in that area. Those community members analyze the results, they analyze the data, and they determine if they agree with the conclusions drawn by the original group of scientists. And if they don't, they provide better feedback so that the next round of idea testing is more precise and more accurate. And then finally, there's the interaction between what science discovers and benefits to society, or just outcomes, influences on society. Testing ideas, especially that have to do with things like medicine, health, diet, nutrition, those new ideas have a very direct impact on society, on just regular people. And how people implement those ideas generates new information that allows us to either refine our understanding of the ideas that we thought we knew, or it causes us to ask more questions, which leads to more exploration and discovery. Sometimes, too, our community analysis and feedback triggers new questions to be asked, which again feeds exploration and discovery, or the community at large determines new and interesting ways that those scientific ideas can benefit people. Okay? So there's a lot of interplay and a lot of interactions here. Science is not an isolated field. It's also not a field where individuals could work in isolation. You have to submit your ideas to review by others, and those ideas then are released into the world where they can have impacts. We're going to go through each of these sections of science one at a time. I want to focus first on exploration. Science starts with the exploration of new ideas. Sometimes it's the exploration of new places on the planet, the exploration of new organisms. But to explore your world is to ask questions. Especially ask questions about things that you observe in your surroundings. Like, I wonder why an animal sleeps during the day instead of at night. I wonder why another animal only eats that particular fruit and not any fruit it's presented with. The observations watching an animal eat, observing when an animal sleeps, are what lead you to ask these questions. Okay? Now, there's a lot of observations you can make. Depending on what observations interest you the most or inspire you the most, that's what helps you to decide what specifically you want to explore about the world around you. Okay? But observation here is key. Everything starts with this, noticing what's happening in the world around you and then asking questions about it. Once you have settled on a question, it's time to test it. Try to figure out what the answer to that question is. And that's where the core of the scientific process comes into play, this idea of testing ideas. To test our ideas, we use something called the scientific method. Now, this is probably the scientific process that you have been taught before in school, but you see here it's embedded in the middle of a much larger and much more complex set of ideas. We often oversimplify science and say that, well, the scientific method is science. It's not. It's one tool we use as scientists to help us interpret our data and, even more importantly, how we gather our data in the first place. We use the scientific method to first identify a question we want to ask, then come up with educated guesses about what that question's answer might be, and then we determine how we want to test those educated guesses to see if we're correct or not. All of this is a process of gathering data that will help us draw conclusions about our question. Here's a rough overview of how the scientific method works. Notice again where we start is with asking a question. We are going to make observations, and based on what we notice in our environment, what we see, we are going to ask questions about what we've observed. But again, observation is key here. Now, this may look a little bit different than the scientific method you've been shown before. There might be a couple of extra steps here. After we ask a question about something we've observed, our first step is actually to do background research. We go out and see what's already been found about this particular question. It's important to make sure that the question we're asking hasn't already been asked and researched. We want to see what's already known about our animal or about our system to make sure we're asking a unique, new question, one that hasn't been addressed before. We also might find out information that allows us to answer a more specific question or a better informed version of our question. Either way, doing our background research is what allows us to be very precise and accurate in the questions that we ask. Now, once we understand a bit more about our animal or about our question, thanks to our background research, we construct what's called a hypothesis. A hypothesis is either a statement that tries to answer our scientific question, so in other words, an educated guess, or your hypothesis can also be a question that your experiment is going to try to answer. So for example, if I'm interested in why rats are active during the day or during the night and not during the day, I could either form a statement-based hypothesis or a statement-type hypothesis, which would be an educated guess, something like rats are active at night to avoid predators that are active during the day. That would be a statement that tries to answer our scientific question. But we could also format a hypothesis like this. We could say that, or we could ask, are rats more likely to be active at night if their predators are active during the day? You can see how both examples are testable statements. In our first example, we can design an experiment that will either support our statement, support our hypothesis, or fail to support it, which would suggest that we need to come up with a new hypothesis. Our second example, we can design an experiment that can answer this question. We can set up an experiment that will determine whether rats are more likely to be active during the night if there are predators, or if they are equally likely to be active at night whether or not there are predators. Okay. After we've constructed our hypothesis, we'll design an experiment to test it. And we'll talk about that experimental design in the next couple of slides. That experiment is going to yield data, results. We then have to analyze those results and see if we can draw conclusions from them. See if we can interpret what they're telling us. Now there's two possibilities that, two possible outcomes that can come from that analysis. The first is that the data supports our hypothesis. Some people say it means your hypothesis is true. I'm going to cross out true because it feels like an absolute statement. Your hypothesis is supported. I'm picky about that because a future experiment could disprove our hypothesis, meaning it's not actually true. Saying the hypothesis is supported indicates that you recognize that while your data supports your hypothesis, future data might not, and that's okay. If you find you can support your hypothesis, you'll write up your results and report them to the community. But what if you analyze your data and you find that your hypothesis is not false, it's not supported? Okay. So your hypothesis is actually either not supported or maybe it's just partially supported. Well, if that's the case, you still report your results. Check that out. Even a rejected hypothesis, an unsupported hypothesis, and the data that goes along with it is an interesting result. Even negative results are informative, and they tell you something that future researchers might be able to use. So you report your results no matter what, so the community can build their knowledge off of them. But you don't just report your results, you also go back to the drawing board. You think about what that negative result means in the context of your research, and then you try again. Construct a new hypothesis and go through the steps one more time. And that is the scientific method. One thing to keep in mind with the scientific method is that you're always testing two hypotheses. Always. You only tend to write out one, though. The hypotheses that we're used to working with are these ones down here, the alternative hypothesis. It's represented as H sub A, or just HA. The alternative hypothesis indicates that there is something interesting going on. So there is something different from, Let's call it normal. Okay. This could mean that there's a relationship between the two factors you're interested in. It could mean there's cause and effect at work. It could mean a number of different things. All right, let me rewrite that. There we go. Yeah. So saying, for example, that rats are more active at night in order to avoid their predators during the day, that's an alternative hypothesis. But it's the alternative version of what? It's an alternative to what's called the null hypothesis, H sub naught. This is also called the hypothesis of no effect. It means that there's no difference, nothing's happening, there's no relationship, there's no correlation, there's no cause and effect. Essentially, the null hypothesis says that what you think is happening isn't. Everything's boring, everything's normal, everything's business as usual. Rats are not active at night in order to avoid their predators during the day. Rats are active at night for some other reason. Or maybe rats aren't more active at night at all. The null hypothesis indicates that the relationship you think exists doesn't. Any alternative to that, any relationship that does exist, any cause and effect that does exist, all of those are alternatives to H naught. So we call them the alternative hypotheses. I'll give you examples of what this looks like. We'll talk about H naught and HA in the context of experimentation. Experiments are tests under very controlled conditions that are designed to answer specific questions. Specifically, they're designed to assess whether or not your hypothesis can be supported. There are two major types of experiments: the manipulative and the observational. Manipulative experiments yield the strongest evidence. They're tests that are designed to determine if changing one factor, and only one, in a system affects the other parts of that system. So for example, let's say you've got these two greenhouses. Each greenhouse has the exact same amount of lighting. They have the exact same what's called a photo period, meaning how much daylight you have per day. Each one gets 12 hours of light per day. They have the same number of plants. There are 3 plants in each of these trays for a total of 3 times 3, 9 plants. Okay. Same thing down here. 3 times 3, 9 plants. Okay. I'm going to tell you a few other things about this system. These plants receive the same amount of water, regardless of greenhouse. They receive the same nutrients, too. Oops, sorry. Notice that the information I'm giving you is all information that would affect the growth of a plant. Plants need a certain amount of light each day. They need a certain amount of water. They need a certain combination of nutrients. And these are all the same type of plant in both greenhouses. Okay. The environment in each greenhouse, in other words, is the same. All the things that plants need to grow, including, say, CO2, carbon dioxide, they're the same in both greenhouses. The greenhouses are basically identical, except for one thing. The temperature that they're kept at is different. Notice that this greenhouse down below has a higher temperature than the greenhouse up above. One question that you could answer with these greenhouses is whether temperature has an effect on plant growth. Your hypothesis, HA, might be that higher temperatures will result in faster growth. That would be your alternative hypothesis, and you'd be able to test that using these greenhouses. After you placed your plants, you would measure all of them every day for, say, 2 weeks, and then at the end of that measurement period, you would see if the plants in your second greenhouse that was warmer grew larger than the plants in your first greenhouse. If they did, that means you can accept HA and reject the other hypothesis, H naught. The null hypothesis in this case would be that temperature has no effect on plant growth. Remember, H naught is always that there's nothing happening. There's no relationship, no correlation, no interesting pattern. H naught in this case would be that temperature's got nothing to do with your plant growth. If you don't see a difference in plant size between your two greenhouses at the end of that 2-week period, then you have to reject HA and accept H naught. Now, manipulative experiments require that things called variables be regulated and controlled. So variables are aspects of the environment that can change, and those changes might affect the result of your experiment. That's why you want to control them, which just means prevent them from changing. All those factors that I listed as being the same between our two greenhouses, all of those were variables. Temperature was the one that we were actually interested in. But there were other aspects of the environment in these greenhouses that could change, like the amount of water the plants received, the amount of CO2 they received, the amount of light that they received. There were a number of different things that could have been changed in these greenhouses. We wanted to control everything except for the variable that we were interested in. And here's why. Let's say we changed the temperature in our second greenhouse to make it higher than the first. But let's say we also gave our second greenhouse more water. Now, if that was the case, and we saw bigger plants in our second greenhouse, we couldn't actually say that that was because of the increase in temperature. We couldn't say that confidently, because we also had an increase in water. Plants need water to grow too. It could have just as easily been the increase in water that caused increased growth, or, even more complicated, it could have been both. Maybe you needed both an increase in temperature and an increase in water to see greater growth. Maybe one or the other isn't enough, and you need them at the same time in combination. This is why we try to only test for the effect of one variable at a time. Otherwise, it makes interpreting our results much more complicated because we don't know exactly which variable caused the changes we're seeing. Manipulative experiments generate stronger data, stronger results, because they eliminate all possibilities except for one. Okay? They control every possible variable except for the one that you're interested in. The reason we're able to do this with manipulative experiments is usually they aren't done out in the wild. They're done in places like these greenhouses or in laboratories. In other words, really controlled settings that are often a little bit artificial. Manipulative experiments don't tend to reflect real-world complexity for this reason. They can sometimes be oversimplified. The other problem is lots of really interesting variables can't actually be manipulated. They're too big and they're out of our control. Think of things like weather or tides. Too big and uncontrollable. Much more complicated than what we're able to recreate in an artificial environment as well. So there are limitations to manipulative experiments. That's where observational experiments come into play. These experiments don't involve manipulating anything. You show up someplace natural, somewhere in the world, and you observe. And you compare what you're seeing to other locations or other environments that have similar conditions, and then attempt to draw conclusions from the comparison. These observational experiments are valuable because they show real-world complexity. Okay. By comparing natural systems with no manipulation involved, you tend to see true patterns as they would exist in nature. Okay. So for example, let's say you're interested in the diversity of insects that you find in grassy fields. Maybe you're wondering if you get different insects in some parts of the world than you get in others. Okay. So maybe you go in South Central California. You find a grassy field at sea level with mountains along one edge. And then you start censusing all of the insects that you find in this grassy field. Okay. And maybe you find there's a fair diversity of insects present. Maybe you find 1,000 species of insect. That's kind of a lot, but it's possible. Now let's say you're interested to see if latitude has an effect on insect diversity in these environments. So you fly north up to Seattle, way up here. Okay. You find an almost identical field. It's at sea level, it's grassy, it's got mountains in the background, it's as similar to your original field as you're able to get. And you start censusing insects again. But this time you find that maybe there's the same number of insects, but there's a lot less diversity. They all tend to be the same type. Okay. Maybe you only find 400 species at this higher latitude. Okay. You have controlled as many variables as you can. It's the same type of environment. It's got the same basic geography. But you see that there is a difference. You have fewer species at the higher latitude than you do at the lower latitude. What you need to do next is try to explain why. It could easily be that that change in latitude also results in a change in climate. It's probably colder further north than it is closer to the equator. Temperature is probably hotter down here. Insects like warmer weather. You might get more rain at this higher latitude. Maybe it's drier further south. Could also be that the amount of light during the day is less at the higher latitude because their days are shorter during the winter. And there's more light during more parts of the year, longer portion of the year, the closer you are to the equator. All of these things could affect how many different types of insects you find. You have found a true difference, but the trick is it's harder to explain that difference because there are so many variables that you simply cannot control. That's the trick with observational experiments. These comparative experiments occur in open environments where you can't control most of your variables. Means your results tend to be more complicated and sometimes harder to explain. You can't point to a single variable and say that's the cause. But they also tend to reflect what's actually going on in the environment, which is very valuable in and of itself. If you want to compare the pros and cons of the two experimental types, note that both of them test the validity of a hypothesis, so they'll help you to either accept or reject your alternative hypothesis. Manipulative experiments are going to yield the strongest evidence, since most of your variables can be controlled. But remember, a lot of things simply cannot be manipulated, and they're also not going to show you real-world complexity. Observational experiments will capture that real-world complexity, but the conditions in nature aren't neat and clean. So your results are rarely easy to explain, they're rarely simple and straightforward. How do you choose which one to use? It just depends on the question that you're asking. Before we can talk about how you design an experiment, there's some experimental terminology we need to go over because we'll be using it in context. The letter N is used to represent your sample size, or the number of actual measurements you've taken, your number of true data points. The more data you have, the better. The more data you have, the more likely the conclusions you're going to draw will represent reality. Now the trick is, these data points have to be what's called independent. They can't influence one another. So for example, if you are interested in how big the leaves grow on a certain species of plant, and you collect the data like this, you go and find an individual plant, and then you measure its leaves. And you find that the average leaf size is 4 inches. You can't say that for this type of plant, leaf sizes are generally 4 inches. Why not? You only measured one individual plant. What if that individual plant has weirdly large leaves? You collected 3 pieces of data here. 1, 2, 3. But the problem is, they weren't independent from each other. They were all related. They were all influenced by the same factors. In this case, they're all part of the same individual. They all received the same amount of nutrients. They all grew under the same conditions. What their sizes tell you is how big leaves grow for that individual plant. If you want to know how big leaves grow for this type of plant, you've got to go out and find a whole bunch of other individuals. They're the same species, but maybe some have grown under easier or better conditions than others. Maybe some have slightly different genetics than others that might influence size. But if you want to get an average measurement for leaf size across all individuals of this type of plant, you've got to collect data, leaf measurements, from different individuals. Each of these leaf measurements, this one, and this one, they're all independent. They're completely different plants. The size of one leaf is not at all related to the size of the next or the next. Okay. Now, a sample is what you collected your actual data measurement from. A sample can be either a single data point, like for example, the size of a leaf, maybe the height of a tree, or it can be a group of related data points, like the widths of 5 leaves from one tree. You can still use your data from this original plant that you measured leaf size from. You would measure all its leaves, you get the average size of leaves for that plant, and then you would compare it to the average sizes of leaves from your other plant. Maybe this plant's average leaf size is 3 inches. Maybe this plant's average leaf size is 4 inches. This one, 3 inches. You'd notice that the average leaf size across all these individuals is between 3 and 4. By measuring multiple leaves per individual plant, you get a better representation of how big leaves are for that plant as a whole. If you only measure one leaf, like over here, you may have measured by accident the smallest leaf on the plant, or the largest. If you want a more representative piece of data, you would measure all of the leaves that are present and average that number. Okay. That would be your sample. Here are your steps to designing an experiment. Your first step is to identify what are called your treatments. Treatments are the variables you're changing to look for an effect, and this is really important. Okay. The treatment in our original experiment with the greenhouses was temperature. Does temperature affect your plant growth? The treatment that one greenhouse received was to have an average temperature, about 20 degrees centigrade. The treatment the other greenhouse received, the true treatment, was to raise the temperature by 5 degrees. What if we were curious about how CO2 affected plant growth instead? Maybe we kept CO2 levels normal or average in one greenhouse, and we raised those CO2 levels in the other. In this case, the treatment would be CO2. Everything else is the same. What if our experiment was observational? These guys down here. Okay? Maybe one question we could ask, one hypothesis, is, do leopards choose prey based on prey size? So what's the treatment? What's the variable we're changing? It's not location, it's not time of day, it's not environment, it's the size of the prey animal. Prey size is our treatment. That's the variable we're going to be looking at, the thing that'll change that we're interested in. Does landscaping affect insect diversity? In this case, landscaping will be the variable. We'll see if unlandscaped wild areas have different numbers of insect species than nicely landscaped garden areas. Your next step is to identify your variables. These are aspects of your environment and the organism that can change. Okay? These are things like temperature, again, CO2, also things like characteristics of the organism itself. Okay? We'll talk more about that, how organisms and their characteristics can change and become variables a little later on in the semester. I'll give you specific examples. Now, the variable that we're really interested in is what's called the response variable. That's the variable that's being measured, that is changing because of your treatment. So your treatment is the variable you're manipulating. Okay? You control the treatment. You control the temperature inside the greenhouse. You control the amount of CO2 the plants are receiving. The response variable is the one that you expect to change in response to your manipulated treatment. In the case of our greenhouses, your response variable would be growth. Now, what if we did this CO2 experiment? What if we changed the amount of CO2 in the greenhouse? What would be the response variable? Well, the treatment is CO2. The response variable is plant growth. That's what you expect to see change. In our leopards example, size was our treatment. Our response in this case is going to be the choices that the leopard makes, if it chooses larger prey or smaller prey more often. Does landscaping affect insect diversity? Landscaping was our treatment. The diversity of the insects is going to be our response. That's what will be different, potentially, whether or not the environment is landscaped. Third thing you have to do is design a setup that's called your control. The control does not receive any treatment, meaning you don't manipulate any of the variables in your control setup. The control is critically important. Critically important. Without a control, you can't draw any conclusions. The reason is you need the control to provide a basis for comparison to see if your treatment actually changes anything. If we didn't have this top greenhouse, and we wanted to test the hypothesis that increased temperature would increase plant growth, we could set up this second greenhouse, pick a high temperature, and grow our plants and then measure them, and maybe say that every plant is or has a height of 50 centimeters. But then what do we do with that number? Okay, all of our plants are 50 centimeters tall. Is that large? Is it small? Is it average? We don't know. We don't have a basis for comparison. By including this top greenhouse with this lower temperature, and by measuring the height of these plants, we might see that these plants, on average, only grow to about, say, 38 centimeters. Knowing that the height of the plants in this control greenhouse is only 38 centimeters, that then allows us to say that the increase in temperature in our treatment greenhouse caused an increase in growth. Without the control, we can't make that statement. So, what would be our controls for these experiments? How does CO2 affect plant growth? Well, we probably want one greenhouse with elevated CO2, and then another greenhouse with average CO2. The one with average CO2 would be the control. What about a new question here? Does grazing by deer affect grassland productivity? Think of productivity as being the amount of plant growth. Well, that's a pretty easy one. If grazing by deer is our treatment, which it is, you would want to have one field where you've got deer. Do it like this. So one field with deer. And then another field without. The one without is your control. And then again, does landscaping affect insect diversity? You would probably have two different plots. One that's wild, no landscaping, and one that's landscaped. The wild one would be your control. Again, the control provides you with a basis of comparison to see if your treatment has an effect, to see if it really does change something. Once you have gathered your data, you've done your experiment, you have followed the scientific method, you're going to have a bunch of numbers or a bunch of results, and you got to figure out what they're telling you. Okay? You have to interpret your data. Okay? You might have data that supports your hypothesis. You might have data that rejects your hypothesis. Your data might also inspire you to come up with a brand new hypothesis. Maybe your data was surprising, wasn't what you expected. Maybe your data is so surprising you have to revise your assumptions, rethink everything about your question, and approach it from a new angle. A single experiment often does lead to a lot more questions, and thus a lot more experiments. And that's how scientists fill their time. They find one result, and it leads them to 5 more questions that give them 5 more results and 15 more questions. And that's how you fill a career doing science. So again, remember this big picture of the scientific process. We've covered testing ideas, and that you get those ideas from exploration, observation, discovery. The next step is to take your ideas and send them out into your community to see if your interpretation matches everyone else's. Community analysis involves other people reviewing your results, discussing them, and then seeing if they can replicate them, seeing if they can come up with the same results you did. During this process of review, you'll frequently come up with brand new questions and ideas that are generated out of this conversation and this back and forth with your peers. If enough studies test your hypothesis and are able to replicate it, meaning repeat your results, and enough people over time re-examine your hypothesis, test it from different angles, and if it is consistently supported over and over again, many, many, many times, your hypothesis might become what's called a theory. A theory is a well-tested and widely accepted explanation of something. Theory is the strongest word we will use in science. We never say something is the truth. We try to avoid saying it's a fact, because everything is open to reinterpretation if better data comes along. Theory is the strongest word we'll use. It's reserved for only the best tested and most supported hypotheses. This constant re-examination and expansion of ideas is really at the heart of scientific approaches to answering questions. There's no such thing as settled science. You hear that term in the news sometimes. It implies that we've stopped asking questions about a certain topic. And that's never true. Scientists are constantly re-examining and re-evaluating even our most well-supported theories, coming at them from new angles, asking new questions, thinking about them in different ways. And it's one of the things that makes science so interesting and so valuable, is we're completely willing to reevaluate our conclusions if better data comes along.